Okay, it looks like we have our participants in. I'd like to welcome you this morning to our trainee research award presentations. I'm Susan Vogelmeyer on behalf of my co-chair, Eric Hung. We are happy to welcome you today to hear talks by Allison Wong and Kristen Rudd. Just to let you know the purpose of the Trainee Research Awards, this is the 9th annual presentation. The purpose is to showcase original research done in the Department of Psychiatry and Behavioral Sciences of advanced trainees in this high visibility forum in our grand rounds. So to remind mentors and mentees that the applications are in general around January, and the presentations are around now. So next year, for those trainees who will be moving on to either their independent career or another phase of their training, please consider applying for the award next year. I'm happy to have our 2 trainees today and remind you that there's another 2 trainees next week who will present their work. Our committee, I'd like to thank the committee that also includes Steve Hinshaw, Andrew Crystal, Dave Manoli, Dan Mathalon, and Marina Tula-Shams, as well for choosing these awesome awardees who are going to talk to us about everything from basic science to health services research. And they represent many of our sites, from the VA to Mission Bay to all the way out into the community. So with that, I would just welcome everyone to this, and I'm going to turn it over.
It is wonderful to introduce Dr. Kristen Rudd. She earned her PhD in developmental psychology with a quantitative distinction from UC Riverside, where she was a Chancellor's Distinguished Fellow. And we were thrilled when she joined us here at UCSF in 2019 as a research postdoc at the Center for Health and Community, where she was funded by my NIH ECHO Children's Study Program project grant, as well as the Division of Developmental Medicine. And she came here with the expertise in development of children's physiological stress regulation and its impact on socio-emotional well-being. But during her time here, she's impressed us so much with her intellectual curiosity, her advocacy for those with less opportunity, and her courage and willingness to, really bridge disciplines and extend her work into social stress exposures beginning in pregnancy period, as well as a breadth of child health outcomes. And she's done this work across 4 different study populations and research teams. And despite stretching herself in so many new areas of learning, she impressively published 10 empirical articles while she's been here, 8 of which were first authored. And in addition to this prolific publishing, amidst the chaos of this pandemic, she's continued her passionate commitment to diversity, equity, and inclusion, particularly efforts targeting first-generation college students and those involved in the foster care system. She's currently our postdoc representative serving on the Departmental Diversity Committee and is a mentor in the UCSF-wide First Gen program. Dr. Rudd's excellence across domains in combination with her lovely personality brought her great success on the job market recently, where she garnered several competitive offers and ended up with a top choice, a tenure-track research and teaching position on the faculty at the University of Colorado. And I think her talk here today will help you understand why. And it's been an absolute pleasure co-mentoring Kristen with my former mentee and now esteemed colleague, Dr. Danielle Rubinoff, who will tell you a bit more about why we're so proud to work with Dr. Rudd. Danielle?
Thank you so much. So I first met Kristen well over a decade ago when she was an undergrad RA at a different institution, but when she applied to be a postdoctoral scholar with us at the CHC, I immediately remembered her, which is just a testament to her very lasting impression as just an incredibly smart and driven researcher. And what's particularly special about Kristen is that she not only has the knowledge in the field to really develop these novel research questions that have the power to advance our knowledge, but she has the statistical skills and the leadership and the fantastic writing skills to actually carry these projects to completion. And she's really not deterred by projects that require her to learn new skills or stretch her bounds. And I think you'll see that today. These are areas where she actually really thrives. So she's taken her background in individual physiological systems and really broadened that to continue to consider their interactions in these really complex analytic frameworks. And I know that I speak on behalf of her other mentors at the Center for Health and Community, especially Dr. Eric Prather and Dr. Wendy Mendez, when I say that we are going to miss her terribly when she moves on to this tenure track position, but she is so ready for it. And we are so excited to see her take this next step in her career. So I will turn it over to Dr. Rudd now for her to share her terrific research.
Thank you so much, both of you. That's so kind. And it's nice to reflect on my couple years here so far. And of course, to share all this work with you. So it looks like things look good on that end. So I'm incredibly honored and excited to share my work with you all today. And the work that I'm going to be sharing explores pathways from early life stress to later risk for psychopathology. And just summarizing, some research over the years has really come to recognize that the etiology of psychopathology is really multifaceted and includes influences across psychosocial, biological, and broader environmental factors. And understanding how and why problems arise is important for developing these effective treatment and prevention efforts. And so in particular, experiences of stress and adversity early in life increase risk for developing later psychopathology. However, these pathways towards maladaptation or resilience are really complex, and they depend upon a variety of individual, family, and broader contextual factors. And we really see a lot of variation in whether an individual exposed to adversity develops problems. And so we see some who are protected against this risk, but there's others who may actually be particularly vulnerable after such exposures. And even beyond understanding this individual variation within people, and that which really tells us about what types of social, environmental, and biological factors may help be protective against adversity. Studies looking at the mechanistic pathways from adversity exposure to maladaptation or resilience will help us to not only identify who is at risk and who should receive specific interventions, but also understanding these pathways provides prevention and intervention targets or ways that we can intervene. And specifically identifying early risk phenotypes is really important because if we know who is responding in a dysregulated way, even if they might not be manifesting in a diagnosable psychopathology, we could intervene before disease manifests, which has huge implications for overall health promotion, as well as combating these rising rates of child psychopathology. And so in recognition of the importance of identifying these underlying mechanistic processes for understanding psychopathology, the National Institute of Mental Health established the research domain criteria framework. And this framework emphasizes identifying pathophysiological mechanisms that contribute to the full range of variation in symptomatology. So from typical functioning, everything in between, all the way to psychopathology, while also focusing on these complex multi-level influences across genetic, physiological, social, and environmental domains. However, there's also been recent revelations around this framework that has placed even more emphasis on understanding the broader social context and how this social context either facilitates or combats psychopathology development. And so this is really emphasizing that social experiences such as lifestyle, the neighborhood you live in, and even your exposure to different types of adverse events is really important for identifying these more holistic early phenotypes of risk. And so what this RDoC framework really gives us is a precision medicine approach to prevention. And rather than looking at diagnostic classifications or categories, we're identifying early phenotypes that may manifest in general problems or psychopathology more generally. And in particular, this RDoC framework highlights regulation across both physiological and behavioral or cognitive domains as important potential mechanisms to examine in identifying these transdiagnostic vulnerabilities. And so a great measure to potentially capture these risk phenotypes that has a fairly established developmental progression is executive functioning. And executive functioning encompasses these complex cognitive processes that are really central to children's normative development. And they include aspects such as inhibitory control, working memory, and cognitive flexibility. And all of these are really important as children work to navigate their everyday lives. And in fact, we see that these executive functioning abilities have pretty well documented associations with social interactions, sensation seeking, risk-taking behaviors, academic achievement, and really many more aspects of children's social, emotional, and cognitive processing that are really integral for their healthy functioning. And similarly, we also see that deficits or deviations from the normal progression in executive functioning across early childhood has been linked to a number of psychological disorders across childhood and even into adulthood, which may make it a particularly informative transdiagnostic indicator. And so when we're looking back on these pathways to psychopathology from early adversity, it may be particularly useful to look at executive functioning or other similar measurements or transdiagnostic type of indicators as precursors to psychopathology instead of waiting for problems to develop. And so then evaluating the mechanistic pathways in this pre-symptomatic transdiagnostic type of framework. And so to do this today in the project that I'm going to be sharing, I'll be focusing in on how regulation across two core physiological systems act as a pathway between early adversity and executive functioning, in that it might be a risk for later psychopathology. So the first system I'm going to be focusing on today is the HPA axis, and this is responsible for the neuroendocrine component of the stress response, which is measured via cortisol. And cortisol is a hormone that circulates throughout the body to support these mobilizations for stress actions. And this process of cortisol release is somewhat slower acting than some of the more immediate responding systems. And we see peak cortisol reactivity typically occurring around 30 minutes after a stressor has been introduced. And another really core adaptive system for responding to stress is the autonomic nervous system. And the autonomic nervous system is co-regulated by two complementary branches, which I'm sure you've heard about many times. So we have our sympathetic system, which we often hear about as the fight or flight response. And the opposing side of that system is the parasympathetic system, which many people often refer to as the rest and digest. But the main thing here is that these branches are supposed to, or are theorized to, work together to mobilize these flexible stress reactivity and recovery from challenges. And an interesting piece about the ANS is that it can be measured through a lot of different indicators like hormone markers, pupil dilation, and skin conductance. But I'll be using cardiography today in this study, and cardiography is really nice because it represents a moment-to-moment assessment of these responses where we can look at both branches of the autonomic system on the same type of time scale. And the two primary indicators to do that, so for parasympathetic nervous system, we have respiratory sinus arrhythmia, and this is basically the naturally occurring variation in heart rate that's in tune with respiration. So this gives us a measure of our parasympathetic activation. And for our sympathetic system, we have pre-ejection period, which is a systolic time interval of the duration of a full heart cycle. And so theoretically, the HP axis, as well as both of these branches, should work together to mount our flexible mobilizations to stress. But few studies have empirically examined this, particularly when we're looking at early childhood, where there's a lot of really rapid development between both of these systems that could potentially be very impactful for later functioning. However, as a notable and exciting exception to this, my co-mentors, Nikki Bush, Daniel Rubinoff, and I have recently published two studies characterizing multi-system regulation in young children. So in each of these studies, we identified 3 distinct subgroups of multi-system responses, and these groups had considerable replication across both of these independent samples. And these studies are particularly exciting, not only as the first to identify these subgroups in young children, but also they provide evidence to support some of the theories around the developmental progression of HPA axis reactivity, which we were able to identify as coming online as early as 3 years of age, which is a pretty novel contribution to the field of child development. And so these novel studies taught us a lot about the characterizations and subgrouping of these types of multi-system responses. However, the next natural step was to evaluate whether these profiles function as a pathway for the associations between early adverse experiences and later risk for psychopathology. And so therefore, the current study, we first wanted to evaluate these subgroupings of physiological responding to evaluate profiles of underlying physiological risk. And then putting this into an RDoC framework or utilizing an RDoC approach, we evaluated the associations from early adverse experiences to multi-system regulation to subsequent variations in executive functioning. And we evaluated these aims using Dr. Nikki Bush's longitudinal evaluation of 113 parent-child dyads. And these dyads were enrolled of a study on the influences of environmental stress and maternal weight and health on her offspring's own development and health. And the current sample here was very diverse with respect to racial ethnic background, level of family stress exposure, and socioeconomic background. However, to be included in the study, women's income needed to be 500% below the poverty line, which meant that the majority of families were considered low income, particularly when we think about the cost of living in the Bay Area. So many of these families were low income. And to evaluate the first aim of assessing these multi-system profile characterizations, we utilized a gold standard developmental challenges protocol at age 3. So this included an audio recorded neutral story task, followed by 3 challenging tasks across cognitive, sensory, and emotional domains that produced age-appropriate challenges for these 3-year-olds. During this challenge protocol, we also continuously collected autonomic regulation using spot electrodes that we placed on the children's neck and chest to collect both impedance and respiratory measures. We then created an average rest score across that whole neutral story and an average reactivity across all developmentally challenging tasks. We collected salivary cortisol using a swab, which we placed in the child's mouth for about 30 seconds or until it was saturated. We collected 1 sample before the initiation of the stress protocol, and then the final sample was collected approximately 30 minutes after that protocol ended in order to capture peak stress reactivity. And so to identify or characterize these distinct subgroups of multi-system physiological responses across both resting and reactivity, we use latent profile analysis or latent class analysis. And latent profile analysis is a really useful advanced statistical tool that identifies these heterogeneous clusters or subgroupings of responses across the sample. And this is really a major advantage over traditional techniques that take the average level across everyone and assume that they all have similar responses to these stressor tasks. So using this technique, we were able to identify 3 distinct subgroups of physiological responses at age 3. And first we found one group that included children whose multi-system functioning was characterized by the lowest activation of sympathetic and parasympathetic systems at rest, and then generally average reactivity in response to these challenges. And this is in line with what we've generally considered to be a typically adaptive response across both rest and reactivity. So therefore, we termed this group the active copers mobilizers profile. The next group that emerged was characterized by generally moderate to low activation of systems at rest and accompanied with the highest cortisol reactivity in response to challenge. So we termed this the HP access responders profile. And finally, a third profile was characterized by the highest activation of parasympathetic, sympathetic, and HP access systems at rest. And this was prior to any initiation of the stress protocols. And these children in this profile also didn't mount any additional response to the challenge. So in order to reflect this heightened arousal at rest, when no challenge was really present, we termed this the anticipatory arousal profile. So the next step after characterizing these profiles was really to put them into a test of pathways from early adversity to later executive functioning. And to do this, we utilized Mother's Report of Children's Exposure to Traumatic Events on the TESI, And the TESE is particularly exciting because it's actually created by some superstars here at UCSF. And this is a report of children's exposure to a range of traumatic events. So separation from a primary caregiver, maltreatment, or accidental traumas, such as being in a serious accident that result in an injury. And we also used a standardized tablet-based assessment of children's executive functioning. And this task specifically taps working memory, inhibitory control, and cognitive flexibility. And this task-based performance has been nationally normed based on children's age. So we get a standardized score that combines the child's accuracy and response times with national averages. And to evaluate these longitudinal pathways, I implemented a specialized BCH technique, which basically just allows me to evaluate the associations between children's adversity, their physiological latent profiles, and executive functioning in a path model that's very similar to a mediation framework. So although results indicated that there was no direct association between early adversity and executive functioning, early adversity did significantly predict profile membership, such that children who experienced more type of adverse life events from birth to 18 months were more likely to have an anticipatory arousal multi-system physiological response. And this response, which as a reminder is characterized by these high stress activations when no challenge is present, also predicted executive functioning, such that children who had this type of response at age 3 had the lowest executive functioning scores at age 5. And this was measured, as a reminder again, as a task-based performance, not mother report or anything like that. So this finding is really exciting and novel, as it's really the first study to put these multi-system profiles into a longitudinal evaluation. And our findings are notable, especially given the nature of our sample. And our sample was predominantly lower socioeconomically resourced families, and many of the families are families of color. So our sample on average encounters a pretty high rate of exposure to stressors, and their experiences are traditionally understudied in psychological science. However, despite these compounding risk factors of the sample on average, we're seeing that a lot of children in our sample are not really evidencing much dysfunction. In fact, they're getting along quite well, suggesting that there are some great protective processes at work in this community. And the variability that we're seeing in physiological responses and range of outcomes really provides some hope on our ability to identify effective protective factors that may be particularly salient for children and families from these high stress contexts, and even those who may be evidencing these risky physiological profiles who are on that path towards psychopathology. So potentially this could be a great group and sample to look to for ways to intervene on this pathway towards problems. And so just putting all these findings together, we were able to identify anticipatory arousal response as a potentially pathophysiological mechanism linking early adversity to deficits in executive functioning. And thinking about what this response means, having a higher activation across systems at rest may be a pretty adaptive response or mechanism to manage stress when you're living in an excessively risky context. However, remaining in this activated state when there's no challenge present can lead to some wear and tear on these systems, which may produce some long-term complications. And thinking even beyond our documented findings here with executive functioning, this type of physiological response has also been hypothesized to be associated with early forms of insecure attachment, as well as later anxiety disorders, which further suggests that this may be an early risk phenotype that could be really useful for constructing and implementing intervention efforts. And we also identified some early emergence of HPA axis reactivity. So although only 14% of the sample was characterized by this type of response, this finding, along with our previous 2 studies, which I mentioned earlier, that was in both younger and older children, combined, these suggest that age 3 may be a sensitive period for the development of these responses. And our findings also provide some evidence for and suggest that children's experiences of adversity may actually become biologically embedded within these stress-relevant physiological systems. and that these processes of stress getting under the skin may occur in a manner that influences their later executive functioning, which can ultimately place them at increased risk for developing later psychopathology. And as mentioned, these physiological responses may be useful indicators for targets or indicators and targets for intervention, especially when we're thinking about working with young children who sometimes or maybe often can't verbalize the self-report responses that we're using or that are really helpful in highlighting risk. Looking at these regulatory components that can be directly measured, such as physiological regulation, may give us a more objective look at these underlying processes and provide early risk indicators that aren't influenced by this limitation in speech and even in parent reporting bias. And of course, there's a lot of amazing providers here on this call and at UCSF generally that know some really great tools to help calm kids and help people learn to regulate. And there's also some really interesting and exciting new applications of biological interventions that may be useful tools in the future as those come along more in their evaluation and development. And so the current study that I shared with you all today had a number of unique strengths, which I've mentioned throughout, but we had a longitudinal cohort of racially and ethnically diverse young children, which is a pretty hard population to reach, especially since they're so socioeconomically under-resourced. And we also had multiple assessments of physiological measures across these multiple stress response systems, which provided a lot of really rich data for us to use. However, we also had a number of limitations. So first, although we utilize this gold standard technique for evaluating the antecedents and the consequences of these latent classes, this is not a formal test of mediation. It's also worth noting that the HP Access responders group had a relatively lower proportion of children, which may have made detecting associations in this group a little more difficult. However, this may also be a function of the normal developmental timing, as I mentioned, of these systems kind of coming online. And so in the future, our team and others in general, expanding upon this work, it'd be really great to fully trace out these models to full pathways to actual diagnosis in order to identify additional intervention targets and prevention or protective processes across multiple levels of impact, as this is really an important first step in increasing precision medicine approaches to prevention and intervention. And I just wanted to conclude by thanking my co-mentors and co-authors on helping me through this work, and my lab members for everything they do on the daily basis. And this study is actually published, so there's a QR code if you'd like to link to the full publication. Special thanks to my lab mate Amanda for telling me how to do this. Thank you.
Fabulous presentation, Kristen. It's amazing to see something that I planned so long ago come to life in such a sophisticated and beautiful way. I see lots of clapping. Is there anyone who has a question for Dr. Rudd? Julio, do you want to go? Wait, you're muted. Sorry, Mateus put his hand up digitally, but I saw Julio's visually. Okay.
Thank you. That was a superb talk. I wonder if, I can see how there is a logic, kind of an adaptationist logic to early adverse experiences and the phenotype you found, the high anticipatory, because it would prepare the child to live in a stressful environment. But is there any logic why that phenotype would then connect with executive function deficits?
Yeah, so when we think about executive functioning and cognitive regulation, it's also really intertwined with our physiological regulation and our ability for our physiological systems to either support, our memory and our ability to maintain multiple tasks in our mind. They're all kind of intertwined. So it's a link there that a deficit in our physiology or our physiology being too ramped up could inhibit our children, specifically in this example, ability to take in those working memory cues and process things in a really adaptive way that's needed for a lot of these executive functioning tasks.
Thank you.
Mateus?
Yes, hi. Thank you so much for this talk, Dr. Rudd. That was really amazing. This is great work. And I wanted to ask a question that is kind of related to this last question, but maybe if you could expand a little bit on a possible meaning or possible pathways for the other latent profile, not the, I'm sorry, I forgot how to call them, but not the arousal. Yes, no, the HPA responders, I think. So are there any associations there? Exactly, yeah.
Yeah, that's a great question. And I think I alluded to it a little bit, but I think to expand upon that, we're seeing it kind of emerge at age 3. So in the study I mentioned previously, the 18-month profiles, we don't see much HPA axis reactivity, but we see it at age 3. So it seems that to be developmentally when these responses are kind of coming online. So I would imagine, or we're hypothesizing, that this is a normal developmental progression. So this development of HPA axis reactivity is really important for these kids. So maybe kids who aren't developing that type of reactivity response could be the ones who might be at risk as well. But we need some more longitudinal work to really path this out. And with 14% of the kids, as I mentioned, we might not be able to detect associations there, but hopefully doing more longitudinal work, we'll be able to kind of suss that out and see what the impact of the development of that response or lack of development of that response could mean.
Thank you. I note that we're at time. We're actually a minute over. Congratulations to you, Kristen, and also to Allison for such fabulous presentations. Danielle, did you want to say one thing as we wrap up?
Just congratulations to both presenters. They were just examples of exceptional work. And I feel so honored to be in a department with both of you.
Yeah, thank you to the department for recognizing these junior superstars and giving them a chance to shine.
Yes, thank you all so much.
Susan, do you need to take us home, or is that it? Have a wonderful day.